reason we do science is because there's things in the world that we care about and that we want to change and solve. And so like it's really important to surround yourself with people that are supportive of what you want to do and that are there to really chase truth together. The Beckman program gives you that supportive network that gets you really off the ground and gets you started.
Hi, my name is Abigail Fisk.
And I'm Kannan.
And we are members of the class of 2023 Beckman Scholars at The Ohio State University. Today, we are here with Dr. Michael Wolfe. Dr. Wolfe, could you please introduce yourself and tell us what year you were a Beckman Scholar at Ohio State?
Yes, hi, I'm Mike Wolfe. I was a Beckman Scholar in, I believe it was 2011. And I am currently a postdoc at the University of Wisconsin-Madison, studying bacterial transcription.
I was wondering if you would be able to touch a little bit more on the research that you were conducting while you were a Beckman Scholar here at Ohio State.
Yeah, so while I was at Ohio State, I worked with Dr. Jane Jackman in the biochemistry department, studying how different enzymes basically are able to recognize exactly what molecule they need to modify. There's a very important molecule called tRNA that helps translate information from the DNA code to the protein code. We are trying to understand the enzymes that actually modify tRNAs to make them be able to perform that duty, both in bacteria and in human cells.
How was your experience with Dr. Jackman as a mentor, and has that relationship lasted?
Oh, absolutely. I can say without a doubt that Dr. Jackman is the reason that I am a scientist. I mean, was an excellent mentor. Jane was really great about making sure that I had an independent project that I was working on as an undergrad. I had ownership over that project. I was meeting with her personally to talk about that project. And so I really felt like I was another grad student in the lab and I was treated as such. And it was intimidating at first, definitely. I mean, it was really stressful. I had no idea what I was doing. Nobody ever has any idea what they're doing when they're starting out. But she was really gracious with me. She took the time to explain the background behind the methods we were using. I really can only say amazing things about Jane. She truly is the reason I'm in science.
Would you say the Beckman Scholars Program has influenced your career trajectory in any way?
Oh, absolutely. I did a undergraduate engineering degree, and my Beckman Scholars research project was in biochemistry. And it's because of that research experience that I got my PhD in biochemistry. I'm now a postdoc working in biochemistry. In Jane's lab, I did a lot of wet lab in vitro biochemistry. Again, so reductionist, you're taking and breaking things apart. It's like taking a cell, putting it in a blender, and then isolating out the parts that you want. And the thing that you miss with that, you get a lot of detailed information about how those individual parts work, but you don't get a lot of information about how those parts work within a larger system. I got a lot of great biochemical thinking out of Jane's lab. In my grad work, I did all computational. So I spent 5 years basically developing how to learn how to program really well, how to learn how to deal with large data sets. I learned a ton of statistics. And I was really just kind of understanding, okay, I did protein nucleic acid interactions in Jane's lab in a test tube. So now I'm going to do that at a broader scale, at a whole cell scale, looking at many different protein nucleic acid interactions from many different players and trying to understand how that network informs biology. My grad work I did with Lydia Friendolino at the University of Michigan, and now I'm in Dr. Landik's lab at University of Wisconsin-Madison. And so I'm trying to marry those two fields of thought, coming back to the bench, but bringing with me my grad experience to understand how bacteria express their genes and what the consequences of that expression is. And so the main thing that I'm studying is every single time that an RNA molecule is made, it has the potential to make this 3-stranded structure called an R loop. And what that R loop does is it basically, the RNA comes down and re-anneals with the template strand of the DNA. That pushes the non-template strand out into like basically unprotected, and it's really mutagenic for cells. And so there's this balance that needs to be made, and that R loop is sometimes used for doing different processes in the cell, but if you have too many of them, you're going to die because you have too many mutations accumulating. And so understanding how bacteria balance that, we're trying to understand what the players are that help control those R loops while transcription is happening. And we're trying to understand like what the consequences are for the cell for those R loops.
Are there any particular memories that have stuck with you during your time as a Beckman Scholar?
Yeah. I mean, the whole thing was just so fun. It was fun to be able to get into the lab and do experiments, have the support network that you needed where you didn't feel like it was all so overwhelming. I feel like what I really appreciated about the Beckman program was that really strong structure aspect and that community aspect. I've learned so much, and the environment was so positive that it made it fun to go to work. And to many extent, a lot of what an undergraduate degree is learning how to think. But what you don't usually get an opportunity to do as an undergrad is to generate new knowledge. And so what I really enjoyed about my undergraduate research experience is that I was working in a lab, doing experiments, and being really the first person in the world to know something about the world. And then the Beckman Foundation and this Beckman Scholars Program is what made it possible for me to dedicate a substantial amount of my time as an undergraduate to doing that research.